It's recording. So the previous recordings of last year are available on the YouTube channel of the Midwest Grape and Wine Industry Institute. I'm going to put the link to the chat box when I will stop talking. So if you want to take a look at what we provided last year, that can be helpful for you. So feel free to go on the YouTube. Then, as usual, ask your question using either the Q&A box or the chat box. So that's supposed to be on the bottom of your screen. If you click on 3 dots, you may have access to that. If you have any issues, try to reach out to Drew or myself by a way or another. And so all the questions you're going to ask would be answered at the end. So we will have 30 minutes at the end to take care of that. And also you will have access to a survey, a quick survey, just to have your feedback about this session and how we could improve our work. So today, the organization is going to be, we have Mr. Tom Mondor and David Spector from Gusmer Enterprises and Chris Hansen, who will start talking about yeast selection and the practical aspect of yeast selection. And then we're going to have Dr. Anna-Katharine Mansfield from Cornell University, who's going to talk also about yeast selection, but more from a research aspect. And then we have 30 minutes for question and answers. So first, thank you, all of you, to be joining us for this webinar. Thank you, Tom, David, and Anna-Katharine for accepting the invitation to present. And I just leave Drew doing the introduction of our first presenters.
I will. Can you hear me okay?
Yes, all good.
Excellent. Our first presenters, as mentioned, are Tom Mondor and David Spector from Gusmer Enterprises. Tom is the technical sales rep for Gusmer Enterprises in the states of Iowa, Minnesota, and Wisconsin. He holds a diploma of brewing through the Institute of Brewing and Distilling. He was a lead brewer at a regional craft brewery in the U.S. for the better part of a decade, and is a very active member of the MBAA and the ASBC. He's currently pursuing a Master of Science in Brewing and Distilling through the Heriot-Watt University in Edinburgh, Scotland. I'm glad I know how to pronounce Edinburgh. David Spector started out his winemaking career as a 17-year-old looking for a way to access alcohol. After joining a winemaking club in his hometown of Philadelphia, he knew this passion would become a lifelong quest. After pursuing his undergrad degree in economics from Penn State, he completed a handful of vintages around the world and then earned a master's degree in enology from Cornell. Working in a variety of functions from winemaking, quality, Lean Six Sigma, and in sales for large winery groups such as Gallo, Constellation, and Vintage Wine Estates. Those are certainly some of the big boys. Or girls. David has acquired a number of entertaining stories about the wine industry that he may or may not share with you, us. Currently, David works for Chris Hansen and travels the world to assist wineries and breweries with fermentation expertise. When not traveling, he can be found working from home in Venice Beach, California, where he lives with his wife and cat. Today, Tom and David are going to present on the practical uses of yeast. Hopefully they'll make some comments about our high acidity Midwest wine grapes and fermentations as well. Thanks, guys. Off you go.
Thanks, Drew. Let me share my screen here. And please let me know if you can see this. Okay.
It looks good. Yep.
Looks good.
Thank you. Thank you. All right. As Drew said, my name is Tom. I work for Gusmer Enterprises, and I'll be joined today by David Spector of Christian Hansen. And today we're going to be talking about the practical considerations for yeast selection. Today, just a quick overview of what we're going to be going over. I'll just touch on quick, who and what Gusmer is as a company, the yeast overview, just kind of go over yeast in general, and then we'll move on to the cool and cold climate considerations as far as yeast selection goes. And then how to pick out yeast selection or what you're looking for when you're browsing your local vendor's yeast selection, and the different characteristics each of those yeast varieties can bring. And then we'll also just touch quick on a data sheet and a COA example. So when you are looking through the yeast, you know what you're looking at, and then when you receive your yeast as well, to make sure that you are checking the COA and making sure you're getting what you pay for. And then at the end of my presentation, David will take over and talk about the non-Saccharomyces yeast strains available to the winemaker. All right, a quick overview, as I said. Gusmer is a family-owned company, currently in its 3rd generation. We focus primarily on fermentation and filtration products. We have a number of different facilities in California, one in Wisconsin, and one in New Jersey. And the way we operate is direct technical sales reps. As Drew mentioned, I'm currently the regional sales manager for the area, and we like to work one-on-one with our customers and more of a consultative approach as opposed to an Amazon, so to speak, going online and just kind of picking out, filling up your cart and checking out. We like to talk and discuss the process and your products. So that's that. So what are yeasts? So yeasts are eukaryotic microorganisms, part of the fungus kingdom, that are facultative anaerobes. So they can operate both aerobically and anaerobically. Specifically, we prefer in the fermentation realm of the anaerobically, as they will then produce ethanol. They divide themselves into mother and daughter cells, and that's their form of reproduction. They consume sugars, in the case of winemaking, primarily glucose and fructose. They require nitrogen sources, in this case, amino acids and ammonia, as well as vitamins, trace minerals, and oxygen. And for oxygen, well, I did say that they are typically in fermentation anaerobic. They do require oxygen to help synthesize sterols that keep their permeable membranes permeable, so that nutrients and sugars can flow in, and byproducts like ethanol and CO2 and sulfur compounds, things like that, can flow out. Yeast produce, as I touched on already, ethanol, CO2, higher fusel alcohols, esters, sulfur compounds, different acids, aldehydes, and many other byproducts that happen or are created during fermentation. So yeast, obviously, is a very important winemaking decision. It's something that you can research ahead of time. There is a lot of good data out there that can help you pick the yeast for your grapes. Being in the cold climate region, not all of it is geared towards the grapes that we grow around here. I live in Wisconsin, so I know the grapes grown around here are not often well represented in the typical sales book or data sheets, but I think that's changing slowly. But it's not, your selection of wine yeast is not always as important as fruit quality. It's certainly an important factor, but overall, fruit quality typically does come first as far as the wine that you're going to be producing. Something else important to really understand about yeast is that it's one of the few things that a winemaker can be in control of as far as pitch rate and selection and adding nutrients and things like that. Grapes will bring their own natural microflora with them. And so unless, I mean, there are certainly places in the world and domestically here in the US in some areas that will let their grapes just ferment with the microflora that's present. But to get the products that we want, that we desire as winemakers, or if you're making this, similar products year after year, if you want that consistency and that quality, we have to be able to control that yeast factor and outcompete the microflora. And other factors, especially around here, we're not, like cold weather is something that we deal with constantly, whether it's a hailstorm that's going to destroy the vines, or an early frost or a late frost, things like that can certainly change the fruit quality overall, but also destroy the vineyards as well, something that we have to deal with. So yeast influences flavor, body aroma, and acid balance of the wines. And it's also important to match your yeast to your grape varietal and desired style. you can always take a particular yeast that says maybe it's good for making a Riesling, and always expect to make the perfect Riesling. Understanding your grapes and that year-to-year variation as far as sugar, pH, TA, if there's smoke taint, if you've had dry seasons, wet seasons, infections, and being able to address that with your yeast selection will help you make the best possible wine. And it's also important to note that you want to be able to express the best characteristics, but also address the less desirable traits, which some of the native grape varieties, depending on how you feel about it, foxy, musky aromas, things like that. How to downplay some of those and accentuate other more desirable attributes. As I said earlier, winemakers should have a good understanding of their year-to-year vintage variability, but sometimes stuff happens, and it's hard to always count on your grapes being similar year to year. Different vineyards, even in the same area, can have slightly different growing conditions and produce very different grapes. So, but understanding that can help you get the final product that you're after. And climate change too. We're talking about cool and cold climate grapes today, but I was in a fairly long discussion last week with a winemaker who primarily grows cool climate varietals, discussing climate change and how over the next 10, 20, 30, 50 years, how the local climate can change substantially, or has the potential to change it substantially. And that's going to change the kind of grapes that we're growing here, the grapes we're able to grow, the grapes that we are currently growing, and how that's all going to kind of shake out. So why use cultured yeast? It's a known quantity, and when I say that literally, I mean that you know what you're getting. You're getting a 500-gram pack of yeast, dried yeast, typically. Your COA, which we'll touch on later, should have give you an idea of the number of cells that are within that package of yeast, so you know how much to inoculate your wine with, or your must with, and you can track that from year to year and batch to batch, and it's something that you have control of. Cultured yeast that we use, Saccharomyces cerevisiae, it's adapted for winemaking. It operates well at the pHs of typical wine musts, or grape musts, I'm sorry, but also has tolerances for alcohol, 17 up to 20% even, which a lot of the more natural yeasts do not. They will die off before they can reach that high of alcohol content. Predictable fermentation kinetics. You know that when you pitch a certain amount of yeast, you can expect certain fermentation kinetics. So you should know that if you are have good nutrition and good mixing, and you're getting the right amount of yeast cells in there, you should be able to have a good understanding of what your fermentation is going to look like, how long it's going to take, things like that. And you should also know then what kind of organoleptic attributes those yeast are going to provide to your wine. And as I mentioned earlier, it outcompetes the natural microflora that come in on your grapes. So types of yeast to use. David will be touching on some of these other ones a little bit later, but Saccharomyces cerevisiae is the most common cultured yeast for wine and for beer, and for other products as well. But primarily when we're talking about wine yeast, that is the one that we're talking about, is the cerevisiae. So cool and cold climate varietals, not well represented in the literature, but that is changing. I know we at Gusmer are working on a number of different products right now that really will accentuate or help a winemaker in this area of the world choose the product that they want to use, both with yeast, but with also malolactic bacteria. These include, the American varietals as well as the French American hybrids, and then the hybrids largely produced and bred through the U of M program, like Marquette, Frontenac, Brianna, La Crescent, which many of us in this area grow. And as Drew mentioned earlier, grapes grown in this region have their own unique properties that the vinifera from grown out west in the United States don't have. So the Labrusca varieties, known for their foxy, musky attributes. Some people are okay with this, some people aren't. The very high sugar concentrations that we can get out here, the high TA, pH with a question mark, because I've heard many people talk about how pH can start very low, and then depending on the potassium content, by the end of fermentation, it can end up very high. But having a broad pH spectrum when you're looking at what yeast you want to use, something to definitely take note of. And we can get very, very low in this region, and some yeast are able to handle that, and some aren't. And then, as I said earlier, shorter growing season. It can get cold, but as we know, a couple weeks ago, it can also get very hot. But overall, the growing season is relatively shorter until climate change has its say, and we have to kind of deal and work around that. So what differentiates the different wine yeasts? There's as you can see, a whole list of what you might be looking for when you're picking wine yeast. There's some very simple ones that I'm sure everyone's familiar with. You know, the optimal fermentation temp. You know, what you want your tank set at if you have cooling or heating. Cold tolerance around here. If you're doing any late season harvests, you know, and you don't have heating, which I know some places don't, you know, being able to make sure your yeast are staying warm enough. Alcohol tolerance with those high sugar contents, we can make some pretty high alcohol wines, and making sure that the yeast are capable of tolerating it and actually finishing fermentation. And also, once you do get those higher alcohol levels, that the yeast are able to stay healthy too, because if the yeast start lysing, for any reason really, but for alcohol, if they start lysing, you know, you will be releasing some of those unwanted compounds that are typically inside the yeast themselves, like sulfur compounds and others that will start to affect the flavor of your wine. Nutritional requirements. If you can send out or do it yourself, you know, check to see what your yeast assemblable nitrogen levels are in your musts. This will help you dose nutrients. We sell Micro Essentials powder, which is the Gusmer produced product, but there are many out there. Making sure that your yeast are getting the appropriate amount of ammonia and amino acids. And a proper nutrition will equal stronger fermentation, but also a cleaner fermentation, less off flavors, more predictable fermentations. So making sure that you can adequately dose in nutrients is extremely important and shouldn't be overlooked. Glycerol production, SO2, H2S, VA production, polysaccharides, foam production. Well, you guys can read all these. The other one at the bottom there, how does it affect MLB compatibility? Some yeasts have been shown to disagree with malolactic bacteria. There has been a couple of studies on it, but essentially some yeast seem to work better than others with malolactic bacteria. And if you're going to be going through MLF, it's something to definitely pay attention to with your yeast selection. And some yeasts have special properties as well outside the typical norms. So on this list here, I got beta lysase activity or thiol releasing, you know, making sure that, or increasing your aroma. Sometimes thiols, you know, when they're talking about thiols, they're usually referencing pineapple or guava or tropical flavors, things like that. But there are other thiols too that may be disagreeable with the type of wine that you are making. So you might not always want thiol releasing yeast. So it's definitely something to experiment with and see how it reacts. But you know, more aromas, not always a good thing depending on what you're making. Beta glucosidase activity, bioprotection, which David will talk on a little bit. Well, bioprotection in general is using other yeasts or bacteria to protect your product through the process of winemaking without using or not using as much of chemicals and sulfur and things like that. Another important one for this area too, with very high malic acid levels, is some yeast can metabolize malic acid and produce alcohol. So it's something some yeast strains can do, some yeast strains can't. Your salesperson will be able to direct you to those if that's something you're interested in. But typically, not more than 20 or 30% of malic acid can be metabolized through yeast. So it's definitely not going to get rid of all your malic acid, but it can help bring those levels down, helps balance your acids a little bit more. Organic, if you're looking to make organic products or organic yeast on the market, direct vessel inoculation. So direct vessel inoculation would be not having to rehydrate your yeast. It literally will just get dumped into the tank. We do have one on the right there called Jazz, which is a direct vessel inoculation. No H2S production. I was talking about this earlier, but there are products out there that cannot produce H2S through classical breeding. That can be a big help, especially I've worked with a couple winemakers, both in the United States and Canada, who they have a couple of varietals that produce H2S no matter what they do, it seems like. Nutrition, a couple of things we've experimented with, but if you're having some issues like that, finding yeast that doesn't produce it or cannot produce it can be extremely helpful. And polysaccharide production as well. This is just a quick overview, an example of a yeast data sheet. So in the middle, there's the beginning part would be kind of an overview of the different characteristics that you can expect from the yeast typically. And this one in particular, you can see on the right, it's got recommended varietals of grapes, that we don't typically grow around here. But like I said, that is changing, and I think we'll see more and more literature out there really addressing some of the different American hybrid varietals that we're growing. In the middle, sometimes you'll see graphs like this, spider graphs. Otherwise, you'll see bar graphs or things like that that are measuring different byproducts of the fermentation. But for this one in particular, they're looking at some different sensory attributes, organoleptic attributes. And at the bottom is really the technical data, the temperature, cold tolerance, alcohol tolerance, how much nitrogen does it need when you are adding in your nutrients, getting an idea of what yeast might need as far as nutrition. Pillar factor. If you haven't heard of this before, some yeast produce a protein that is toxic to other yeasts and can kill them off. And sometimes that is a desired attribute. Sometimes you want only one yeast in there to produce the wine that you want, and sometimes it's undesired. And I think there's been some stories out there of folks scared of the idea that their yeast might have the killer factor. But in my experience, and please correct me if I'm wrong during the Q&A, it's not a scary thing. It's not going to go out. It's not going to kill all the yeast in your winery or your brewery or anywhere else. But it can actually be a very helpful tool to use if needed. Dosage range conversion factors. Typically, it also give you an idea too of your rehydration methods. Make sure that when you are adding dry yeast, that you're rehydrating it to the manufacturer specifications. Adding nutrients if you need to, adding in must if you need to, but doing that will essentially wake up the yeast, rehydrate them, get them ready for alcoholic fermentation, and give you the best possible viability to go forward with fermentation. And then lastly here, I've got an example COA from one of the yeasts that we sell. If you aren't getting your COAs from any of your products, reach out to whoever it is that you're buying them from and start asking for them. It's really good, useful information to track from batch to batch, year to year. And you can see what you're getting. On this one, it's got the batch number, material number, so you know exactly when it was made, when it's going to be, how long it's good for, but also how many cells are in that pack. And how much bacteria is in that pack, how much E. coli, foreign things like that. So if you're having issues and you're looking to pin down stuff, you've gone through, the temperature was right, you added the right nutrients, you had good oxygen and aeration, you can use this kind of data to go back and really troubleshoot. And as I said, keep it from year to year too, and And if you're not getting it for your yeast or your nutrients or your enzymes, if you're using enzymes or anything like that, make sure you're asking for it and getting a copy. So that's my little spiel on practical considerations for picking out yeast. And David Spector now is going to go through non-Saccharomyces yeast for winemaking.
All right. Thank you, Tom. You can move on to the next slide. So hello, everyone. I appreciate you all taking the time to learn about yeast. You've heard Tom speak about yeast selection, and soon you're going to get to listen to my former graduate advisor present, Dr. Anna-Katharine Mansfield. So luckily for you, I have the shortest part of the presentation, just pretty much a 10-minute primer on non-Saccharomyces yeast. Do you want to hit the next slide, Tom? So you're probably familiar with Christian Hansen. Yeah, there you go. Yeah, that's right. You're probably familiar with Christian Hansen because our range of Enococcus ini bacteria is the industry standard when it comes to malolactic fermentation. My first job out of Cornell was with E&J Gallo Winery, their facility in Livingston, California. That's the largest winery in the world. They're producing more wine there than some countries. Last check was 640,000 tons per year. So we just use Christian Hansen bacteria exclusively there. But today, our focus is on yeast. And for my part, exclusively non-Saccharomyces yeast. And so focus is going to be on the winemaking process before alcoholic fermentation. Now, these non-Saccharomyces yeast can produce ethanol, and they will produce ethanol, but that's not the focus. That's not why you're using these non-Saccharomyces yeast. That's why we're calling it pre-alcoholic fermentation. Next slide. So this is the classical population sequence in winemaking, right? If you're doing a sequential inoculation. Next slide, Tom. And at Christian Hansen and Gusmer, we really view the winemaking process, the biological part of it, in 3 steps. You got the non-Saccharomyces, the rise and the fall of the population there. Then you got Saccharomyces cerevisiae, really takes over pretty quickly. It's just been bred for years to really dominate a wine fermentation. And then finally, if you're going to put your wine through malolactic fermentation, that's when Enococcus ini is the star. Next slide. All right, so at Christian Hansen, today we're just going to focus on the species. I'm not going to utilize our commercial names. The commercial names are there if you need to see it, but what we're more interested today is to talk about what these different types of yeast can bring to your winemaking practices. So we have 4 pure cultures available. That's Lachance thermal tolerance, Pichia claveri, and Taurus Delbrueckii. And we also have some blends, if you can hit the next slide, Tom. The blends are pretty cool because it allows the winemaker to have some convenience. You'll have your non-Saccharomyces there, usually in a higher dose, and then you have your Saccharomyces there, so you don't have to do sequential inoculations for your yeast. Each of the different species has its own features, and that's all right, and benefits depending on the winemaker's needs. But what we're here to talk about is how the different species themselves are going to help you achieve your goals and help you make the best wine possible. Tom, it looks like these next slides might have some transitions, animations. You can just cruise through them till we get to the population sequence. Go back one. Go back one.
Oh, gotcha.
Yeah, the full slide. There we go. All right, so right here we have the sequential inoculation graph. And what I really want you to pay attention to here is the fact that we are going to be inoculating pretty early on. Many of the world's best wines are made with indigenous yeast. So, I live in LA. You can ask all my friends that hang out at natural wine bars. They love wines made with indigenous yeast, indigenous bacteria. But if you want to consistently manage your product, then I certainly recommend inoculating with cultures. And for non-Saccharomyces yeast, you really want to get those microbes inoculated as early as possible. Maybe that's on the harvested grapes. Maybe it's at the crush pad, or maybe it's when you're filling your tank. But you know there's microbes coming in, and Saccharomyces can really take over quite quickly. You might have some non-Saccharomyces yeast or bacteria that are not going to produce the aromas and flavors that your customers want. So I recommend inoculating. Next slide. Might have to cruise through this one a bit. I think there might be 3 animations. One more, one more. Okay, so in our opinion, there are 3 species among all the non-Saccharomyces yeast out there with the most potential for premium winemaking, and that's Taurus delbrueckii, that's Pichia claveri, and that's Lachantia thermotolerans. For those winemakers looking to make an impact with aroma, with flavor, Pichia is really a great species for you to utilize, you know, really upping those esters, upping those terpenes. And I see that my graph is a little bit off-centered, but you get the point. For winemakers looking to produce acid, you know, I know most of us are probably from the Midwest or the East Coast, so we might not have the same problems as winemakers in California bringing in Zinfandel at 30 bricks. You're dealing with high ethanols, you want more acid. Lachantia thermotolerans, that's a really great tool for winemakers, but maybe not for the folks here. And then finally, if you're looking to maximize mouthfeel, then you perhaps want to utilize Taurus delbrueckii. I know when I'm tasting wines, it's just a lot of great wines. Let's take Chardonnay, for example. Flavors there, but the mouthfeel, the texture sometimes can be missing. Yeast can be one tool that the winemaker can use to really complement their wine. Maybe some leaf stirring. There's oftentimes no silver bullet, but often you want to use a number of tools. And don't forget how companies culture their yeast. You know, we're not making these in a lab. You can't do that. Those are all of our yeast and bacteria in the U.S. They have to come from wine. They have to come from must. They have to come from grapes. So we go out, we find wineries or universities with yeast strains that have unique characteristics that consistently produce them over time. And then we isolate them, test it, and then we provide that culture to winemakers around the world. Next slide. All right, so we're going to have to go really quick through this because I do not want to take any time from Anna-Katharine Mansfield. You might have to invite me back, and we'll have to take each of these different species one-on-one, but I'll quickly go through them. Taurus delbrueckii. What are some of the features? Mouthfeel, complexity, lower VA. Next slide. The mouthfeel is coming from that polysaccharide release. That's adding texture. That's adding viscosity. And the longer that you have Taurus delbrueckii, you know, in there, in your wine without Saccharomyces, because Saccharomyces will take over pretty quickly, the more polysaccharides you're going to have released. Next slide. One of the reasons that winemakers are really liking Prelude Taurus delbrueckii in cold climate varieties and cold climate regions is for ice wine, perhaps. It's really osmotolerant. So if you have a high sugar concentration, you can really utilize Prelude. It's not going to produce, you know, a lot of VA or fatty acids. It will produce some ethanol, consume some of those sugars, and really create a nice environment for your Saccharomyces cerevisiae. Next slide. Next slide. Bioprotection. That's the biggest buzzword that I'm hearing today. all of these species will produce some bioprotection for your wines. That is not exactly known why. We don't have a pure mode of action that's going to tell you exactly how it's working. Some people believe that it's the different non-Saccharomyces out competing the bad non-Saccharomyces that might be producing off aromas or off flavors. They're also consuming oxygen. They might be competing for nutrients. Tom talked about killer factor. So there's a few reasons why these products might be protecting your wine. But we have some studies showing that these non-Saccharomyces yeasts, when used as a pure culture at the recommended dose, really do provide some bioprotection. Next slide. Complexity. We talked about mouthfeel, especially in Chardonnay. That is the winemaker's grape, right? You are going to provide a lot of the aromas, the flavors to Chardonnay. And I think the most important part of that wine is the texture. And you can really utilize this yeast to help you out there. All right, so that is the summary for Prelude. We're going to cruise through Fruitzen. Next slide. All right, so Picky Claverie for wine. This one's interesting because it comes in a frozen format. It produces a lot of flavor. If you have this wine and you're culturing it, you can smell banana right from the plates. It's just producing a lot of aromas, a lot of flavors. We isolated it from New Zealand because producers there really wanted to have a Sauvignon Blanc that has great aroma impact. But I'm seeing winemakers utilize it in red wines, Pinot Noir, Cabernet Franc, could be of some interest to Midwest winemakers. Next slide. Okay, that's just interesting to look at. Comes in a frozen block. You add it directly to your wine. Next slide. Oxidation. This is where you can really create some whites, some rosés, and really utilize less SO2 in the beginning of your winemaking process. Next slide. Okay, next slide.
Oops, sorry, you want me to go back?
That's okay. Yeah, the summary is good. All right, so if you're looking to boost some aromas, you want to protect your wine from oxidation, you want to lower your SO2, definitely check out Fruitzen. Next slide, we're going to go into LaShawn.
But in real quick here and say that for folks who are bringing grapes from out west somewhere, adding this in, having your grape growers on that side of the process, add this in before they ship it to you, whether you're getting whole cluster or you're getting juice. Adding that in early and then sending it over can help reduce the amount of SO2 they need to use and help protect the grapes that you're getting too. So
yeah. And we're going to cruise through, LaShawn. I'm sorry that I have to go fast, but I don't want to take anyone's presentation time. So
just 1 minute
LaShawn,
remaining.
1 minute remaining. I can do
Right.
it. All right. So LaShawn thermotolerance. This is really a great use if you're looking to improve your acid balance. They're taking sugar and it's turning it into lactic acid. Next slide. Next slide. Yep, so this is what we're talking about here. You're producing lactic acid from sugar. Perhaps if you are looking to lower your ethanol content, it's not going to lower your ethanol in your wine by 1-2%, but it might give you 0.5%. It can really freshen up your wine. It can be used with a number of other practices to really create a very balanced, fresh wine. And you'll see through some of the compounds that it's producing. For red wines with Concerto, produce a lot of nice berry aromas. And then we're also utilizing our newest strain, just came out this year, called Octave, for rosé production, for white wine production. Okay, next slide. Next slide. Let's cruise to the end. We'll just do a summary because we only have 1 minute left, and we'll allow Anna-Katharine to present. I know most people are quite excited to hear. from her. So, yep, 2 Lachance thermal tolerance. We have now, excellent. We have 4 different non-Saccharomyces. That's the largest range that I'm aware of. But, I'll be honest with you, most of the trials conducted are with varieties in Europe and in California. Those are the regions that a lot of suppliers, a lot of the largest universities are focused on, right? So my request to you is work with Tom, utilize these products in your grape varieties, in your wines, and let's learn together, how to optimize this new frontier on maximizing the aromas and the flavors and the styles of wine that are going to make the Midwest and East Coast wines unique. And I do think that they are competing with California wines, with European wines, and that competition is only going to get more fierce. So I'm looking forward to learning from all of you winemakers out there and working with you this harvest and future harvest. Thank you.
Thank you very much, Tom and David. That was great. Just to have all the tools and the knowledge about the product that you can provide, and that can be helpful for a lot of you, I think. So please remember to chat or to use the chat box and the Q&A box for any question you have for Tom and David about the yeast selection, the practical aspect. And we already have some questions, but that will be answered after the presentation of Dr. Mansfield. So Anna-Katharine Mansfield is an associate professor of enology at Cornell's New York State Agricultural Experiment Station, known as Cornell Agritech in Geneva, New York. She first worked in the wine industry in North Carolina's Yadkin Valley, received graduate degrees at Virginia Tech and the University of Minnesota, and served as the first enology project leader at the University of Minnesota from 2001 to 2008. Mansfield returned to the East Coast in 2009 and currently focuses her efforts in aiding small regional wineries through enology extension, wine sensory evaluation, and research on hybrid wine phenolics and fermentation nutrition. Most recently, and that's effective July 1st, so right now, along with her continuing professional duties, Dr. Mansfield is an associate director of Cornell Agritech. So Dr. Mansfield, the floor is going to be yours. I think you will be able to share your screen. Oh, we don't hear you. Nope. Maybe unplug. Oh, yeah.
Now?
Yeah.
Okay. I have too many buttons. I've got 3 places I can mute myself. Apparently I hit one of them. Anyway, thank you for the kind introduction. And yes, I almost was running late because I was in a meeting about like air conditioning in one of our buildings. So that's the kind of thing I'm thinking about these days, which is, what associate directors do apparently. Let me share my screen so you can see my slides. And Aude, I will trust you to tell me if you are seeing all of them and not some other part of my screen.
Yeah, all of it. If you can make it as a full screen.
There you go.
Perfect.
Looks great.
Okay, wonderful. Thanks to you both. And thanks to Tom and David for those great presentations. I'm going to probably reinforce some of what they say, but that's good. They say that you only remember 20% of what you are taught. And so if the 20% that we overlap on is the important stuff, then we're doing the right thing. So I will do that. So Aude originally asked me to revisit some work that we had done several years ago, back 2013, 2014, at the beginning of the Northern Grapes Project, when we were doing some yeast trials for several of the cold hardy grapes. And I rebelled a little bit because what we found out doing those yeast trials was that it was in some ways more important to understand the reasons that we made the decisions choose the individual yeast rather than just giving you pages and pages of charts of here are the yeasts we tried, these are the tasting notes, these are the chemical numbers, blah, 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 because it turns out that those weren't as important perhaps as some other things. So I am going to give you a talk on sort of what I love best, which is flavor chemistry, and how you can use a little bit of flavor chemistry knowledge to guide your yeast choices. So let's see if I can figure out how to advance my slides. Here we go. But first, pop quiz. What does an aromatic wine have in common with stinky cheese and toe jam? If you know the answer to that, you can put it in the chat. I can't actually see the chat right now, but I will be very interested in seeing what your thoughts are. So think about that, and I will tell you in a little while. But that's your starter question to see if you're paying attention. And as Tom said, I want to absolutely reinforce the things he said. What you need to do before you choose yeast is know what questions to ask. Of course, we know this anytime we're making wine, right? But very important questions are, how ripe are your grapes? How clean are your grapes? Because the most magical yeast in the world will not make up for bad quality grapes or grapes that have too much rot in them. We don't have yeast that fantastic yet. I'm sure that you guys are going to keep working on developing those strains. But right now, your quality is clean grapes, ripe grapes are going to be your key to quality. After that, are your grapes aromatic or neutral? What's your yan? Are you barrel aging? How good's your temperature control? Can you actually control the fermentation, or is there a possibility that it's going to get too hot? If you don't have good temp control, you need to think about maximum temperature, things like that. These are all very important, but the one that is often most interesting to people and most interesting to me right at the moment is, what's your desired wine style? So I'd really like to frame it around that. What style are you going for? And backtrack from there to what are the aromatic components that are important. So if your style is young and fruity, which is probably the fastest thing we're going to make, right? We're going to do a stainless steel fermentation. It's going to be pretty quick, and then we're going to get it out on the market, turn that Frontenac Rosé right around and get it out in a few months or so. Sort of the basic things you need to think about in terms of capabilities your yeast need to have, or it needs to be cold tolerant, right? We're probably going to do a cool fermentation, probably stainless steel, although who knows, maybe something else, maybe glass, maybe concrete. So you want to check those lower temperatures on that lovely chart that I believe David or Tom 1 showed us, the chart that give you all those the details, the COA. You also probably want a rapid fermenter. You want to get this done, and you don't want a lot of yeast lysing. You don't want a lot of release of the inner contents of the yeast that are going to give you creamy and aged biscuity characteristics. We really want to go with fresh and fruity. And you might want a high glycerol producer. All yeast produce some amount of glycerol, but if you want to get something out pretty quickly, but you want it to have some body, checking the glycerol production on those yeast can definitely give you some body. And with viscosity, because of the way our mind and our nervous system are connected, higher viscosity gives you an increased perception of sweetness. So even without residual sugar, having more glycerol gives your consumer a perception of a little bit more sweetness to boost that fruit. And we certainly have yeasts that are able to do that. And then lastly, probably the most important thing if you're going for a young fruity wine is that you want high ester production. There are a whole lot of different esters, or all kinds of chemical compounds in there that are in the ester family. It's a very large and varied group, but they are responsible for almost all of the fruity and floral aromas that you are going to find in your fresh young wines. Some of them come from the grape, but the ones that we're bringing in the grape have minimal sensory impact. They are present in all grapes. They don't characterize any of them. So there's no varietal typicity that you're getting from the grape in terms of esters. There are other compounds, yes, but there aren't sort of esters that typify any particular grape. So in ester production especially, our yeast is the most important factor. And this is where I'm going to get a little bit into the chemistry, but I think it's important because it helps you understand, actually sort of biochemistry. It will help you understand how to optimize your ester production. Esters are synthesized from fatty acids, so otherwise known as lipids, which are in fruit. Sounds funny to think that you've got lipids in fruit, but they are there. And when you get fatty acids plus ethanol, you get ethyl esters, which produce a lot of these fruity characteristics that you like. Cherry and banana are the ones, you know, that come off fresh and early, but also apple and a score of other fruits, pretty much anything except for the tropical fruits like passion fruit and mango, which are a different class of compounds, but your sort of your fruit basket fruits are all esters. And how they form in yeast? Well, those fatty acids form differently depending on the temperature that you are using for fermentation. So if you have warm temperatures and more aerobic conditions, which we usually don't have in wine fermentation, but which can happen depending on how you're treating your wine, you're going to produce longer chain fatty acids. So you're going to produce very, relatively speaking, long chains of these fatty acids and esters. And the aroma of the long chain fatty acids is waxy and fatty. That's why I've got this Johnson wax here. It really can give you kind of floor wax or some kinds of glue. Not necessarily very pleasant. They don't have a big aroma impact in wine. If they're there at all, they often are giving you sort of a background or base that sort of sits down underneath everything else. Unless you've got a really bad fermentation and you don't have a lot of fruit coming from other places. But if we have cool anaerobic conditions, which is what we're going for with young wines that we want to get out pretty quickly, right? We're producing smaller chains. We're producing mid-chain fatty acids, or MCFA. And these MCFAs are quite aromatic and there are varied. There are a whole lot of them and all different kinds of configurations, different kinds of branching on the chains, and all of these minor variations will give you different aromas and flavors. That is to say, it's going to be a mix. You're never going to be able to, at least, I take that back since I've got two yeast vendors on the call with me. At this point, we are not able generally to specify exactly which fatty acids we're going to get. It's going to be a mix. But it's still going to be pleasant and nice and fruity. And it's going to vary depending on which yeast that you use. So here's the answer to our question. Now, the catch is that mid-chain fatty acids on their own, when they are first formed, smell like Limburger cheese, other, I would say aromatic cheeses, but I am a fan of stinky cheeses. And also, believe it or not, they are formed between your toes when you haven't been taking your mom's advice and washing your feet well enough. So toe jam and stinky cheese, the typical aromas of those are from mid-chain fatty acids. Why aren't those in our wine? Well, because fortunately we also have ethanol in our wine. And when ethanol interacts with these fatty acids, it esterifies them. That makes them much stronger in aroma, but also pleasant, happily. So in fact, isovaleric acid, which can smell pretty gross, moldy, sweaty, it is there in Limburger cheese if you have experienced that aroma sensation. When it interacts with alcohol, which you can see both in this chemical representation and down here in our little model, gives you an apple and strawberry aroma. So hooray for ethanol for more reasons than one, but definitely for our the pleasure of our noses. It gives us our lovely esterified fatty acids. So one thing that I do want to mention, I think I did not put it in here. I did not. One thing I want to mention before I go forward is that, you know, we often are told that the reason that we want to ferment aromatic wines cool is because we don't want to risk the loss of volatiles through entrainment with carbon dioxide. The idea is kind of that those aroma compounds will get bound up with the CO2 and they'll blow out the top. And if it's very hot, we'll blow out more CO2 and we'll lose more of those volatile compounds. That's not the case. We now know from a biochemistry perspective that what's actually happening is that if the fermentation is too warm, we are producing less of those mid-chain fatty acids and more of the long chain. So we're keeping the wine cool for that first step, the fatty acid production, not because we're potentially losing things at the end of the fermentation. All right, so esters are definitely important in anything that we want to be aromatic. And esters do have a shelf life. They will over time, they will dissipate. Really, they'll degrade and turn into other aroma compounds that aren't quite as potent. And unfortunately, for those of us who are working in cool climate areas, those esters will degrade more rapidly with lower pH. And so when we have higher acids, we get more of that ester cleavage, and that fruity freshness goes a little bit quicker. So when it would, say, in a, I don't know, in a Riesling produced in a cool but not cold climate, if it would take 18 months to lose all your esters and start to develop aging notes, it would probably happen in as short as, say, 6 to 12 months in a low pH, say, Frontenac Gris. So think about that when you're thinking about your market and how quickly you want to get your wine sold, and what kind of flavor changes you might get. All right, so moving on from esters, what about varietal typicity? What about the other things that make a specific wine taste like Sauvignon Blanc or make it identifiable as a Riesling? Well, any of the aromatic cultivars, and I will include Sauvignon Blanc among aromatic cultivars because even though it's not sort of floral, it does have a very distinct aroma. They've got both free volatile compounds, so compounds that are free to move through the air and move to your nose so you can smell them, and bound volatile compounds. And about 10% of them are free, which means when you are just pouring the juice, pouring the must into the fermenter, about 10% of them are available there, but about 90% of them are bound up to sugars. And some of those will be released during fermentation as the sugar is lost, but a lot of them are bound to sort of longer chain sugars and other compounds that aren't acted on during fermentation. So what we need are the yeasts that act on these precursors, these bound compounds that don't smell, to break them off, the sugars and other things they're bound to, and release those volatiles so that we can smell them, because that's a whole lot of stuff that we might be smelling that we can't. We're never going to have 100% cleavage, but we can certainly do better than 10%. Let's talk about cleavage of specific compounds. Monoterpenes are one that, after esters, you are most likely to be familiar with. Monoterpenes are the lovely floral and fruity and lychee. That's a lychee nut if you've never seen one in the wild or in a can. They give us nice floral compounds. Linalool, which is pictured there on the left, is one of the most common. It's got a very nice perfumey rose-like aroma. There are about 40 different monoterpenes that arise in aromatic grapes, and 90% of them are bound. They're bound as glycosides, which means they are cleaved to, or they're joined to something else. Monoterpenes are not produced by yeast. Unlike esters, which are almost completely produced by yeast, the monoterpenes are produced in the grape. So what we use the yeast for is to release these bound components. If somebody tells you that they have a yeast that is making monoterpenes, they either have, I'm either really behind in my science, or they are confused as to what they're using. So cultivars that we know have monoterpenes: Riesling, Gewürztraminer, any muscat type, any wine that has muscat after the name, muscat, or beginning of the name, Muscat Attenelle, and any of those. And we have found them in Le Crescent as well. And so yeast that say they can cleave monoterpenes would be useful in Le Crescent, because you do have a pool of, or potentially, depending on how it's grown, you have a pool of monoterpenes that can be acted on. If there are no monoterpenes in the grape to begin with, no bound precursors, you're not going to get them, even if the yeast can cleave them, because there's no precursor there to produce that compound. This is a really great schematic that I found from a nice review of precursors in grapes. So monoterpenes are hydrolyzed during fermentation, which means that they cut off that sugar and release it so that it is a free volatile that you can smell. And depending on the yeast and the pool of precursors available, they can hydrolyze between 5 and 30%. We haven't seen any sort of natural fermentations that give you more than 30%. So we were at maybe 10% free monoterpenes. Maximum we can get is about 40%, but that's often enough. I'm sure you've all had Gewürztraminer that knocks you over with rose smell. So we probably don't need to go for 100%. So this just gives you an idea. It's a two-step process where we get some initial pieces pulled off so that the sugar-bound precursor can be acted on. That's removed, and then you can get a free compound there. When you see yeast that are listed as recommended for muscat or recommended for Gewürztraminer, or which say that they can produce floral aromas, rose aromas, that usually is code for these hydrolyzed monoterpenes. I would recommend, if you ever have questions about that, talk to your yeast vendors. Tom and David both said that they really like to have one-on-one discussions, and I would say absolutely use that. They know their products better than anybody else. So if you're unsure whether a yeast that is described as being aromatic is producing esters or is potentially releasing monoterpenes, call them and ask, because they know that mechanism. It's often not listed because a lot of people just don't want to know. But if you know that, then you'll know whether, if it's an ester producer, it's going to give you floral, fruity things regardless. But if it's a monoterpene hydrolyzer, it's only going to give you increased aroma if you've got some precursors that can be hydrolyzed. All right. Now let's talk about volatile thiols. As David, oops, sorry. See, I need to go back. I apologize. As David mentioned, volatile thiols can be good or bad. Let's focus on the good ones, because those are the ones we're often interested in. It certainly is typical of Sauvignon Blanc, where you have like the boxwood and the cat pea as being desirable traits. Also, their volatile thiols can give you grapefruit, passion fruit, mango, almost any of the tropical fruits except pineapple. Almost all of those are typified by volatile thiols. So when you think of those sorts of aromas, they are these sulfur compounds. These are grape derived. Unlike the monoterpenes, most of the precursors are found in the skins. And the pool of precursors available can vary quite a bit. We found in Sauvignon Blanc in New Zealand, for instance, that it really is about, there's like a 2-week window during which the volatile thiols peak. And after that, they actually decrease again. They don't just keep going up and up like other flavor precursors potentially can in some grapes. So a lot of it, again, is grape quality. And when you pick the grapes, yeast do not produce volatile thiols either. They release them just like they do, well, not in the same mechanism that they do monoterpenes, but like monoterpenes, if you don't have precursors in the yeast or in the grapes, you're not going to get any volatile thiols, or at least none that give you these lovely descriptors. And 20 to 60% of them come in the skin. So if you want more of it, that's when you start thinking about skin contact time for your white cultivars. Perception threshold is quite low. There isn't a whole lot in there, but you don't need a whole lot. Humans are able to smell these at very small amounts. As anybody who has had a cat who had an accident in the house knows, it can overpower you quite quickly. And of course, Sauvignon Blanc is typified by these aromas. They also are in Riesling and Gewürztraminer, although they are not as important in it. The typicity of those as are the monoterpenes. Some rosés, especially Grenache rosés, and we do have some volatile thiols in Frontenac Gris for certain. So again, this might be a type of cultivar or a type of yeast that you want to try if you're making a Frontenac Gris and want to go very aromatic. Sort of similar to those monoterpenes, you get some cleavage. There are precursors that are released in the must from the skins into the juice in the must, and that is cleaved to produce these volatile thiols that have the varietal character and the aromas that we are looking for. All right, let's talk about some other styles. Oaky and opulent. We're going to turn away, for the most part, from the fruitiness if we are working with oak, because esters, volatile thiols, monoterpenes, definitely esters and monoterpenes are going to get covered up by the oak. There are some nor-isoprenoids that you might want to, which is another fruit aroma, family of fruit volatiles that you might want to have more of, but that's not dictated by yeast. That's dictated by ripeness. So good ripe grapes will give you more fruit if you're going into oak. So you don't need to look for ester producers. You don't need to look for thiol or monoterpene producers. You might want to look for high manoprotein production, because when you have that lees contact and the stirring, the batonnage, high manoprotein will give you more body, more texture, or more of the sort of the creamy mouthfeel that you're looking for in an oak aged product. Some other I'm going to touch on very quickly. Dessert wines, of course, high osmotolerance. Osmotolerance means that it is that the the yeast are comfortable with a high concentration of sugar in the must. Some yeast that will dehydrate and die, but in high osmotolerance yeasts, that sugar is something they can take. They've got a cell wall that can handle it. And since many of our dessert wines start with relatively high sugar, you're going to need that in order to get the fermentation going. If you kill all your yeast right off the bat, that's not going to help. And so your vendors can absolutely help you figure out which those are. Fortified wines also potentially take a special yeast, but kind of opposite. You might want a weak fermenter, especially if you're doing a port style wine where you want to stop the fermentation by adding brandy or neutral spirits. So you might want low alcohol tolerance so that when you add the ethanol, you actually stop the fermentation instead of having it kind of meander along, like limp along and maybe produce some weird off aromas because it's struggling. So unlike dessert wine where you're starting with high sugar must, you might want to start with something weaker and lower tolerance. And of course, malolactic conversion, I won't talk about this in any detail because Tom already covered it, but you do want a yeast strain that plays well with lactic acid bacteria, and there are some that absolutely do not. And I guarantee you that, especially if you're doing sequential inoculations or co-inoculations, excuse me, that your yeast supplier can talk you through that and tell you which to pair together. Don't just pick a random yeast and a random lactic acid bacteria. And again, David took care of this for me too. There are some yeasts that have some malolactic conversion. They, or excuse me, some malic to acid convert or malic to alcohol conversion, or also some that can do some malolactic conversion themselves without having the lactic acid bacteria. So there are lots of fun new products that you can look through there and see if they fit your style. So just briefly, I wanted to tell you about the yeast trials that we did do in 2012 and 2013. It seems like ages ago now. But we had 4 sites, even though there are only 3 stars here. One was at the University of Minnesota. And then we had one here at Cornell in the Finger Lakes, which is not a very cold hardy region, but we do have some folks who are growing Frontenac and some other cold hardy cultivars, including some in our trial vineyards. And then over here on both sides of Lake Champlain, we had a vineyard in Vermont at University of Vermont, and another one, we have actually a test vineyard on the west side of Lake Champlain in New York. So, and that is definitely cold hardy country. They're only able to grow University of Minnesota and other cold hardy cultivars there. So we, with my colleagues that were then at the University of Minnesota, Katie Cook and Nick Smith, we picked several wines or cultivars that were important at the time, Frontenac, Frontenac Gris, Le Crescent, and Marquette. And this is what we were looking for. And with Frontenac, we wanted to try some ester producing yeast and see if we could up the fruit character, because Frontenac's so nice and fruity anyway. We were making rosés for the most part, for various reasons, but I guess I'll just go on the record and saying that now that we have Marquette and some other red producers, I feel like Frontenac is nicest as a rosé because of that huge cherry aroma. Frontenac Gris, we were trying to pull out those thiol-based aromas. With Le Crescent, we were leaning on monoterpene production and just general aromatic impact, so cool fermentations. And with Marquette, we were looking for varietal character. Lots of reds, like Marquette, like Cabernet Sauvignon, like Merlot, don't have a specific aroma that typifies them, like say Sauvignon Blanc does with that thiol, the grapefruitiness. Cabernet Sauvignon, of course, has got the green pepper aroma, but most reds don't have something that specific, and Marquette is one of those. It's more a blend of different volatile compounds that are important rather than one specific one. So we are just looking for general fruitiness, mouthfeel, and sort of extraction body. So these are the yeasts that we tried. And if you look along, if you look along the top row, you'll see that we use ICV GRE for both of the reds and the Lalvin DB10 for both of the whites. Those are our controls because they are reliable fermenters and ones that we know work. And you can see the choices that we made here. This is kind of all I'm going to show you. I will leave these to you and let you try them, because the short answer to what was best was all of them. Everything worked. Everything fermented to dryness. We didn't get bad H2S in any of these. And this is across all of our, winemaking, we're doing lots of small batches, and which is really, really hard to do. So it's easy to mess up. and we didn't have any trouble there. And when we took these to various audiences, both in the Midwest and in the Northeast, so Vermont and New York, and did sensory evaluations both with winemakers and with consumers, what we found was that there was no one wine that was preferred. No one yeast gave us a stellar wine, or at least a wine that was so head and shoulders above the rest that everybody said, you should use that yeast forever. So what we kind of came to the conclusion of is that all of these work, and it really comes down to the winemaker to decide what your stylistic goals are. And I feel like if you have the data that I just gave you about, how to think about sensory characteristics, and the data that you just heard from David and Tom, and that you can get from other yeast vendors that you are working with, you probably have the tools you need to make your own stylistic decisions. You know what your fruit tastes like coming in from the vineyard. You know how ripe it is and how clean it is and what style you want, and you can go from there. I would rather leave you with that kind of information than to give you pages and pages of radar charts showing you, oh, well, the elixir gave us a little bit more of, the peach aroma, because that may not work for you. It really depends on where you are and what your fruit looks like. The one thing that we saw that was quite pronounced was that the Northeast folks really preferred the Marquette that was made in Vermont, and the Minnesotans really preferred the Frontenac Gris that was made in Minnesota, because we were sending all the wines everywhere, so everybody tasted the same set of wines. But those were both, because we had made them in different locations, those were both very clearly terroir-driven preferences. I think it may have just been that it's hard to taste Marquettes from Vermont if you live in Minnesota, and vice versa. So I think people were used to their own regional style, which is also interesting because these grapes are fairly new. And if we can start developing regional styles so that we can say, oh, a Vermont Marquette is like this and a North Dakota Marquette is like that, I think that adds some interest and gives us some things to talk about when we're marketing our wines and talking to other winemakers. So I think I will leave you there. I do want to thank these folks. Katie Cook now works with Scott Labs, but she was the project leader at the University of Minnesota when these wines were made, or her wines were made, at least. We made some at Cornell as well. And Nick Smith is now at the University of Wisconsin, but here he is in action in a place that might look very familiar to you, Drew. And then just wanted to thank you for your time. This is where I am in the Finger Lakes. And if you want to follow our lab, we do have a Facebook page. You can check in and see what we're doing. We offer online courses from time to time, especially in this post-COVID time, and you are welcome to join us for those if you'd like. So
great. Thank you so much, Anna-Katharine. That was great, especially when you mentioned that even if you use different yeast, the preference of the consumer is what would be driving your winemaking process, I think.
Absolutely. The consumer or the producer, right? Depending.
Or the producer, yeah. Yep. I agree.
I say go with the consumer.
Yeah. Good. Thank you so much. So now it's the time for you to type any question you may have in the Q&A box or the chat box. I realize that we already have some questions. And I think one of the questions, Anna-Katharine, you started to answer it. But I'm just going to say it for everybody, just to be aware of that question. So the question was preferred yeast strains for Marquette to minimize acidity. And so Bill asked this question, and Anna-Katharine said, Bill, when we were running early yeast trials on the cultivar, we liked RC212 and BM45. That was years ago, however. Drew may have found something he likes better by now. So Drew, if you have a specific yeast strains for Marquette.
Well, I have a few things to say. Of course, RC212 is a wonderful, probably one of the greatest Pinot Noir yeasts. And as Marquette is in many ways related to Pinot Noir, it's a great choice. And yes, I know commercial winemakers still using that. BM45, also a great choice, but there's now, I don't, if you've heard of the BM 4x4, which is there you go, Anna-Katharine, Dr. Mansfield. This is a hybrid, blended yeast that is similar to BM 45. It may be partially derived from that. Now, that all being said, all the research marquette, experimental and research marquette that we make at the University of Minnesota, my hands are still tied by using our workhorse D254 yeast. What can you say? D254 is probably one of the greatest red wine yeasts, workhorse yeasts ever. So that's always a great red wine yeast to have around in general. And of course, if you've got Marquette that's a little slightly higher in acidity or lower in pH that may have had to have been picked early, 71B is still a good choice. And it also is one of those yeasts that can reduce your total acidity by about 35 or 40%. So there's my addition to Anna-Katharine's comments. Yeah, great question.
Thank you, Drew. We have another one. So Bill also asked that. He's a hobby grower and have found so far that for non-Saccharomyces yeast to suppress unwanted microbial growth are only sold in large quantities for commercial growers' winters. But I only need a 5-gram packet or 2. How can I find those quantities?
Yeah, so Tom, I don't know if you're supplying those products yet to sort of the home brewer or home winemaker market, where they come in 5-gram packs.
We do sell things to some of the home winemaking stores and kits and things like that, but we don't sell the smaller package individually. It's usually, if they are repacking it, they're doing it themselves and putting it in smaller packs. But right now, the smallest that we have is 500 grams.
And while we're on the subject of buying things from Chris Hansen and Gusmer, how do people get a hold of you to order any of these products or to get a catalog?
Thanks, Drew. You can go to our website, gusmerwine.com, and you can click on Find a Rep there, and it'll take you to, if you just type in where you're located, it'll tell you who your technical service rep is, and customer service number is there as well, and you can just call the number and place an order. I cover Iowa, Minnesota, and Wisconsin, so if you're in one of those states, it's probably me, and my email is, I can put it in the chat here, but if you just need to reach out and just to chat too about stuff, we're good for that too. So,
if I can chime in there, can you hear me?
Yep.
Oh, great. Sorry. After last time, I'm not sure if I can chime in. I mean, I know there are certainly they're very active home winemaker clubs around here that will sometimes go in as a group and buy a big pack and split it up themselves. And if you do that carefully, I'm gonna ask Tom, his thoughts on this when I finish. But if you do that carefully and you store, reseal it in a sealed plastic bag, or if you've got a vacuum sealer at home, do that. Stick it in the fridge. It can last a few years. It will be less viable each year, but it's not going to go completely dead in 12 months. So you could use it for a few years. I don't know the shelf life on these products, Tom. Am I going well outside what you recommend?
No, they're typically shelf life usually about 2 years. But yeah, if you can, aseptically as possible, transfer into smaller packages, and then seal it up and freeze it or put in the cooler, that works the best.
And keep in mind, don't miss the simple math that 2 years can be 3 vintages, three vintages of use in a row. That is to say, 2 years between the first and the third. So I've used second and third year as well, and as Anna-Katharine says, maybe not as viable, but worked fine.
Yep. We have another question, and I think that's a good question. Like, can you use SO2 pre-inoculation if adding non-Saccharomyces yeast?
For speaking from the products that I represent, which the answer is yes. Most of them do have a tolerance to SO2, typically up to 30 parts per million or 45 parts per million. So I assume that's probably pretty similar across the board. when you look at this stuff coming in naturally, those don't always have the same tolerances, which is why we add in SO2. So.
Yeah, and I would like to add that it's not because some yeast produce naturally SO2 that you don't need to protect your wine in another way.
Yeah, good point. Absolutely.
Yes, still use good winemaking practices. All of the extension people will tell you that.
Yes.
I have a quick question, Aude.
Yeah.
Dr. Mansfield, you mentioned cool fermentations or moderately cool fermentations. Can you give me can you give us how you define that for white wines versus red wines? What is a cool fermentation?
You want me to go first? He asked you first, Aude.
No. That was for you.
Oh, that was for me.
Dr.
Sorry.
Mansfield. Yep.
Cool fermentation 55, moderate 65, red wine fermentations and ton and like big ton fermenters can get up to 95. That's too hot. You can kill some yeast. So that's my definition. But Tom may have, they probably have dialed in a little bit more for their product. So I'm going very vague with that.
No, I think vague is good. Yeah. 55, 65 degrees is definitely on the cool side. And keep it under, when my son gets old enough to drive, keep it under 90 if you can.
All righty.
I don't think we have more questions for the moment, but I do have one question for you, Tom and David. Do you know if some yeast have specific enzyme activities that you didn't mention? Like some enzymes, you mentioned that they would, or like some yeast would be able to transform malic acid into lactic acid, but also sugar into alcohol and lactic acid. Yeah. But do you know if some yeast have other enzyme activities that can be used for winemaking purpose on those grapes?
Yes, the answer is yes. So someone just asked me this the other day. It's not that these yeasts are packaged with enzymes. It's that the yeast themselves do have enzymatic side activities. So briefly, I think it was in one of my slides, beta glucosidase activity, beta lyase activity. These will, give you break down enzymatically and give you some different products. Typically, aromatic products is what we're looking for in these processes. So some of them can produce desired flavors and aromas. Some of them, as I mentioned, can give you some flavors and aromas that may be not stylistically appropriate for the wine you're making. So it's important to check out exactly what they have. So, and we sell them, other people do too. So just whoever you're talking with about it, make sure you ask them about it, and if it's something you want to use for those different wine styles.
Yep, good. Thank you. Drew, if you have any questions on your side, feel free to ask.
I wanted to know if either, well, actually, no, one quick pointed question for Tom regarding the Melody product. It says that it acts both as a pre-fermentation and a fermentation yeast. Was that right?
Melody?
Yes, sir.
Where did you see that? I was just curious.
It's the name of one of it was in your
No,
presentation.
I know. I meant what you mentioned about it. The fermentation and was that in one of David's slides?
Oh, may have been in one of David's. Maybe I'm confused. It is the last.
Okay. So Melody is one of the blended products, which I think is maybe where the confusion is. So by blended, it has some of those non-Saccharomyces yeast strains in it, but it also carries a regular Saccharomyces. So it's kind of like co-inoculation, right? So you're adding it all in at one time. You get a little bit of those activities of the non-sac, but then the regular Saccharomyces takes over pretty quickly. So it's kind of like an all-in-one kind of product.
So it's basically it does its work during the Saccharomyces yeast phase.
Yep, exactly.
And I wanted to ask you both Dr. Mansfield and you, Tom. There's some discussions going on. I hear them from time to time about regarding malolactic fermentation, the pros and or cons, the do's and don'ts, or risks or rewards of co-fermenting or sequential fermentation of yeast primary and secondary.
So I'm not sure if I'm going to answer the question correctly, but let me know if it's correct. But you're asking the differences between the two, or the pros and cons of doing it either way, or?
I'm wondering if you have any opinion from your experience or what you hear from your customers regarding doing separate yeast fermentation followed by MLF, or doing them at the same time, co-fermenting them.
Sure. So I think it's going to be probably dependent on the wine in itself, right? So each malolactic bacteria is going to have a different range of pH, temperature, SO2 that it can tolerate, right, to be able to finish out and do its job. So if you have during fermentation, if your pHs are starting to dip below like 3 down into the 2s, which can happen by the end of fermentation, I would or sometimes they can go higher, right? And you can have some issues that way as well. I would definitely recommend co-inoculation at some point, because it's gonna be less stressful for the malolactic bacteria. One, it won't have to deal with as much alcohol because they are relatively alcohol intolerant compared to some bacteria or some yeast. It's also going to have less of pH issues. And then if you're getting later in the year too, and you're worried about finishing your wine all the way through malolactic, you know, they do have some pretty rigid restrictions on temperature, right? So some folks will inoculate and they'll put their wine to sleep all winter. And then malolactic will just kind of drag on and on and on. And then spring comes and the cellar warms up a little bit, and all of a sudden malolactic will finish out, right? So if you want to try to get that done, you know, prior to that, it's and you're under the gun for timing, it can also be very helpful to do co-inoculation or bump it up a little bit, right? So that the malolactic starts off a little earlier.
Awesome, thanks. Dr. Mansfield, do you have any comments about sequential versus co-fermenting?
Yeah, I think I would agree with what Tom said. I mean, I think there are pros and cons both ways, right? But stylistic goals are something to think about. You will get less of the creamy, buttery diacetyl if you do fermentation. You'll get, if you're aiming for retaining more fruit but dropping some of the acid, the co-fermentation is the way to go. And if you are looking for that creamy, buttery, you know, barrel fermented Chardonnay kind of character, that's, you want to do the sequential. But there are difficulties and risks both ways, and definitely if you're going to do co-fermentation, talk to your vendor. Make sure you know which products you're using.
Awesome. That great. Thank you both for your answers. That's some good guidance, which I can pass on to my stakeholders.
And just before we close this webinar, so we have another good question from Eric, who is looking for information about how to design a new yeast program. So what would be the best way to go about the yeast selection process? Looking through catalogs or going straight to the company reps? That's great question, I think. What would be the first step to do? Just calling you, Tom and David, or just trying to duplicate something that has been done?
This is what Tom said, so this is what you have to do. I definitely, I mean, I think the wine industry, depending on the area, can be very open. So talking to fellow winemakers, joining some of the different groups available. I know there's a couple here in Minnesota and in Wisconsin that they talk about these things. Getting an understanding of what people are already doing and their experiences with them can be very helpful. looking online too. Number of the different websites do have like ratings that you can give and some feedback. Reddit is a good place too to get some additional information from anonymous internet users, but it can give you some perspective. But yeah, then reaching out to the different sales reps and seeing if they have any information to share as well. And then also the schools in the area like Cornell and the U of M and Iowa State.
Yep. Good. I just have a quick question, more for my knowledge. Anna-Katharine, did you look at the mouthfeel characteristics in the wines, the Cold Hardy red wines you tried for the Northern Grape Project? So Frontenac and Marquette, did you look at the mouthfeel obtained, or you were just focusing on aroma compounds?
We were just focusing on aroma compounds. I mean, those grapes were so new that, you know, we barely knew how to deal with the acid. So we were starting with acid, and then we were going to yeast, and we were just starting to look at Yan about the time that I left. So mouthfeel was kind of a fancy extra at that point. So oh, do you have work to do? I'm excited to find out what you do. Because there's interesting stuff there, clearly, in terms of protein binding and yeah. Good question.
With phenolic compounds.
Oh, yeah.
How, the finished mouthfeel could be.
Yes.
Good. All right. So we are just 2 minutes after the time, but so thank you very much to all our presenters today, and thank you for the attendees. I know that a lot are gone already, so if you have any question that we didn't answer or any other question, feel free to reach out to all of the presenters or Drew and myself through the email address you have on the slides. Remember that before closing the webinar, so maybe you have received an email with a link for a survey. If you didn't receive the link, you can scan the QR code and you should have access to the survey. And then that's just the list of the future research and winemaking webinar of the series that are going to happen this year. So the next one is going to be August 3rd on microbial spoilage. And I wanted to also mention that there is a town hall meeting provided by a lot of different extension programs and universities from the Midwest and the East US. So feel free to submit your question, and that's really a meeting with a lot of different extension specialists and faculties that can help you to answer all your questions from viticulture, but also enology. So feel free to join this town hall meeting. And on that, thank you so much, everyone. Have a good day.
Thanks, everyone.
Thank
Good to see you.
Thank you.
Bye.
Thank you, Aude. Thanks, everybody. Bye-bye.
Thank you. Bye.